Primarily going to focus on the adaptive, also known as the acquired immune response. Okay? So in chapter 14, we talked about the innate immune response. These are the guys that respond very quickly, but kind of in a dumb way, right? So every single pathogen is kind of the same kind of mild response. Well, today we're going to focus on the guys who will come in to the rescue if, for whatever reason, the innate immune response is not enough. Then we're also going to talk about immunization, which is how we can potentially manipulate the immune system to give us protection without actually getting sick. So, as I said, we're going to talk about the acquired, also known as the adaptive immune response. And this immune response deals with the concept known as immunocompetence, or essentially the ability for you to A, fight off an infection until you're healthy again. And then once you fight off that infection, develop something called immunological memory, which then prevents you from getting that exact same infection again, or at least for a second. To do this, you need to develop both B and T cells, also known as lymphocytes. Okay? B cells produce antibodies, and T cells allow for cell-mediated immunity, or essentially making sure everyone is doing their job as they should. And these T cells are very specialized. In fact, there are thousands of millions of these cells floating around the body, and each one of them responds to a unique antigen. And by having all of these cells, you potentially have an immune response to all antigens you could be exposed to. Okay? Now, with that said, these cells then are very, very specific. So we talked about the innate cells, and we said they have kind of a broad defense. They respond to cells in a very similar way again and again and again and again. The adaptive cells, on the other hand, are extremely, extremely specific. Okay? Not only do they look just for E. coli, they can look for just one specific strain of E. coli. Okay? So very, very specific in nature. And they also have the ability to develop something called immunological memory. And this is the concept where if you get infected with one pathogen, and then you clear that pathogen, it is very unlikely for you to become infected with that pathogen again. Okay? Now, there are some exceptions to that rule. The pathogen needs to not change itself, okay? So it needs to kind of stay the same. And also, it has to be the exact same pathogen. If it's a closely related pathogen, you may not have that protection. So, to do this, there are 5 steps that we're going to talk about today. Lymphocyte development and differentiation. This is the creation of the T and B cells, okay? And this process happens in all individuals, okay, regardless of infection, shortly after birth and then up until about puberty age, okay? Then we'll talk about how the pathogen actually gets presented to these cells so they can become activated. This is a process of challenging B and T cells. And then when B cells become activated, as I mentioned before, they will make antibodies. And these antibodies will bind and kind of remove the pathogen. And then T cells will do one of 2 things. T cells either help the immune response, okay, so they kind of act as helpers, or they can actually kill cells that are abnormal, okay, so cancer cells, viral cells, things of that nature, okay. And the combination of having antibodies as well as cells that can help and then kill abnormal or weird material results in eventual clearance of the infection. So for this adaptive immune response to work, you need to have both B and T cells working together. Okay? So your book likes to call this the dual system, okay, the adaptive immune response. B cells and T cells work together. In fact, they actually communicate a lot with each other. And they need each other to completely clear the infection, at least appropriately. Okay? So there are instances where people do not have enough B cells, do not have enough T cells, and that generally makes their immune system much weaker than someone who has the same amount or equal levels. Okay? Now, as we said before, with all immune cells, they need to be able to communicate with each other very well. Otherwise, they have no idea what's going on. Okay? Because these cells are scattered throughout the body, and for them to really fight the infection well, you need millions of them kind of working at the same time, so they have to be able to communicate. And they primarily communicate by releasing chemicals, they are proteins known as cytokines. So, lymphocyte development, again, this occurs in all individuals from puberty or from birth up until about puberty. And then you actually get infected with something. Once you get infected with something, the antigens on that pathogen have to be processed and presented to both B cells and T cells. When B cells become activated, you get antibodies, and when T cells become activated, you get cell-mediated immunity, or that helping the immune response and potentially getting rid of any abnormal or virally infected cells. Now this is a nice little summary slide that kind of explains everything that occurs in the adaptive immune response. I'm not going to go through it right now because it's just going to confuse you. I will go through it at the end. Okay, so once we go through all the major points, we'll come back and we'll kind of summarize it by going through this bit by bit. So let's first talk about lymphocyte development. As I said, lymphocyte development is a normal kind of growth process. It occurs in all people. Okay, regardless if they get infected with something or not. Okay? This occurs shortly after birth, and then it kind of wanes off for the most part after puberty. Okay? You still will develop T and B cells as you age, okay, after puberty. It's just a much, much smaller amount than what you were developing in your adolescence. They all come from the basic stem cell, a hematopoietic stem cell, which are produced via hematopoiesis in the bone marrow, just like we talked about in chapter 14. Okay. Now, once these T or B cells are produced in the bone marrow, they're not finished. They have an additional kind of training step, which we call maturation. Okay. This is where the B and T cells are, you kind of select for the ones that are going to respond to foreign antigens and not your own antigens. Okay. For B cells, this maturation process occurs in the bone marrow. And for T cells, this maturation process occurs in the thymus. Okay? So it's relatively easy to remember. B, bone marrow, T, thymus. Okay? It's not what the B and T stand for, but it makes the point. Okay? Then once these cells are matured in either of these sites, they then are released into the periphery. And what that means is we just let them go into the lymphatic system or the bloodstream, and then they circulate around in your body literally for the rest of your life. Okay? And they're circulating, waiting for you to get infected. And once you get infected, that's when they become activated and start doing their jobs. Okay? So this whole process is just making the B and T cells and then training them so that they are appropriate. Okay? That they fight the right types of antigens. Now for these B and T cells, and in particular T cells, to function properly, they not only need to be able to recognize antigen, okay, or foreign substances that can cause disease, they also need to be able to recognize a special protein on the surface of cells. Okay? And this protein is known as MHC, or major histocompatibility complex. Okay? This is a receptor, so it's a protein. It's found on many cells in the body. Okay? And this is the way that T cells actually see antigen. Okay? So we talked about in chapter 14, neutrophils and macrophages can go up to antigen, they can just swallow it up and get rid of it, right? T cells cannot do that. They cannot see antigen floating around them, okay? The only way they can see antigen is if it's stuck in a molecule on the surface of the cell, okay? And that molecule is MHC. Now in humans, it's often called HLA, or human leukocyte antigen, okay? That's just the human name for it. For all other mammals, we call it MHC, okay, or major histocompatibility complex. Now, this major histocompatibility complex, or HLA in humans, comes in basically two flavors, class 1 and class 2. Okay? Class 1 is found in all nucleated cells in the body. So every cell in your body that has a nucleus will also have this MHC class 1 molecule. This molecule is designed to present or show antigen to T cells from internal pathogens. Okay? So internal or intracellular, I should say, pathogens. So things like viruses, intracellular bacteria, or even cancer cells or abnormal growth. Okay? And that's why you find it on all nucleated cells, because these are the cells that are susceptible to these types of infections and abnormal growths. Class 2 MHC is found only on antigen-presenting cells, or what we call APCs. All antigen-presenting cells are phagocytes, like macrophages, dendritic cells, and B cells in some cases, that can phagocytose things from the environment and put it in the MHC molecule. Okay? So class 1 is found on all nucleated cells. Class 2 is found only on antigen-presenting cells. Would you expect to see class 1 on antigen-presenting cells? Like a macrophage. So this is kind of a trick question. Class 1 is found on all nucleated cells. Okay? Pretty much every cell in your body is nucleated, including antigen-presenting cells. So antigen-presenting cells actually have both of these molecules on their surface. I'm bringing that up now because it will make more sense later, okay, when we start talking about how these things work. So this is what MHC looks like. We have MHC class 1, MHC class 2, again found on all nucleated cells, found only on antigen-presenting cells. Both of these have little grooves in them where antigen can sit. Okay? So if I'm an antigen-presenting cell, I gobble up bacteria, I break it up into little bits and pieces or little antigens, and I stick it right here in that molecule, and then I just kind of wave it out for T cells to see. Okay? It's the only way that T cells can see their antigen. If your antigen-presenting cells do not have this, or you do not have antigen-presenting cells, your T cells will never become activated, okay? And you'll be immunodeficient. Now, not only do they have to be able to recognize these MHC molecules, they also need to be able to recognize their antigen. Okay, so there's two things that these cells have to do. See antigen, but also see these receptors. Okay? Now, B cells have a receptor called the B cell receptor, and T cells have a receptor called the T cell receptor. Okay, so very easy nomenclature. The B cell receptor looks a lot like an antibody, so this guy can actually grab onto antigen floating around. T cells, on the other hand, cannot, and remember, they have to use the MHC molecule to see their antigen. So what exactly is an antigen? We talked a little bit about this before, but antigen is anything that stimulates an immune response. Okay? And these are often very, very small little pieces of protein. So it's important to note that each pathogen potentially carries with it thousands and thousands of antigens. Okay? Because each pathogen is made up of many proteins. When those proteins get broken up, they then are processed into little pieces, and each one of those could be a potential antigen. So when you really look at it, there are literally millions upon millions of antigen possibilities out there. Okay? So from one E. coli bacteria, you could have 5,000, 6,000 different antigens. Okay? Now, the thing that tends to confuse a lot of people is the way the immune system works, especially with the B and T cells, is that in general, you only have one B or T cell that can respond to one particular antigen. And when you have then millions upon millions of B and T cells, you then have a large amount of cells, or what we call a repertoire, which can then respond to all known antigens available. Okay? So you've got a million, let's say you have a billion T cells. Each one of those responds to a different unique antigen. And when you add all of that up, now you have enough cells in there that can respond to all antigens you may be exposed to. So, T cells, so the T cell themselves, as I said, has a T cell receptor. And this T cell receptor has an extra bit added to it, called a coreceptor. And this can be either CD4 or CD8. Okay? CD4 are found on special T cells known as helper cells. And these are the cells that go around helping the immune response, okay? Making sure that all of the immune cells are functioning properly. Now, CD4 binds to MHC class 2. So that means T helper cells can only see antigen on antigen-presenting cells, okay? Because remember, class 2 is found only on these APCs. CD8 binds to MHC class 1. And CD8 cells are what we call cytotoxic. These are the cells that will destroy any abnormal or virally infected cells in the body. Okay? And that makes sense because MHC class 1 is found on all nucleated cells, and these are the cells that may actually develop a viral infection or be abnormal. Okay? Now, the fun part. So, how do we get billions of these cells that each respond to their own unique antigen? How exactly does that happen? Well, it happens based on chance. It's completely random. Okay? When you look at the genetic material of B and T cells, they have a series of segments, okay, or little pieces of DNA within their genome. And during their maturation stage, so this is where they're either in the bone marrow of their B cells or in the thymus of their T cells, they will literally randomly just pick a couple of these segments and stick them together. Okay? So you've got 500 segments, it goes through and it says, okay, I'm going to pick these 5 randomly, and that's what they get. So it's like shuffling the cards. And this is happening in each one of these billions of cells that you're producing. Okay? And so at the end of the day, you're going to end up with these cells that have all different responses to all different antigens. Okay? A very kind of generic response to everything. But another problem with this is not only are they going to respond to foreign antigens, like E. coli or a virus, they also could potentially respond to your own antigens. Because this is a completely random process. Okay? So here are those segments we talked about, those 500 different bits and pieces. And this is truncated so that it's easier to see. Okay? And what the cell does is it randomly just goes out like this one, and this one, sticks it together, and now that's the antigen they respond to. So it's completely random, arbitrary, just kind of picking out these gene segments. It's almost like they're mutating their genetic material on purpose. Okay? They're picking segments and getting rid of everything else. Now, as we said, this occurs during the maturation step. Okay? And that's going to happen in the bone marrow for B cells or in the thymus for T cells. Okay? So you make your B and T cells in the bone marrow. They then move through their T cells to the thymus or stay in the bone marrow of their B cells. And then they go and they pick their antigen by this random chance. Okay? They just shuffle the cards and take whatever they get. Once they pick their antigen, you then need to get rid of the cells that are problematic. Okay? The cells that are problematic are going to be the ones that respond to your own antigens. So because this is a completely random process, 50% of the cells will respond to all the foreign bad stuff. The other 50% of the cells are going to respond to bone, or to muscle, or to liver, or to brain. Okay? And obviously we don't want those cells, because if they get out into the body, they will go and they will attack those tissues and destroy them. Okay? So what we need to also do during this maturation process is eliminate those cells that respond to self. How do we do this? Well, there are special antigen-presenting cells found in these tissues, the thymus or the bone marrow, that will present every antigen that you will ever express in your body to these cells. If the cell happens to respond to one of those antigens, it is eliminated and destroyed. Okay? If it doesn't respond to that antigen, that means it responds to something outside of the body. And so they're allowed to survive. Can anyone guess what would happen if, for whatever reason, these cells aren't destroyed properly?
They start destroying.
They start destroying the body, and what's that called? What happens when your immune system starts to destroy something in your body? Autoimmune. So this is how autoimmune diseases develop. Okay? For whatever reason, the guys that respond to your own antigens don't get destroyed. And if they then go attack the islets of Langerhans in the pancreas, then you have type 1 diabetes. Okay? They go and attack the myelin sheath, you may get multiple sclerosis. Now, after this process, after this maturation, you are left with a whole bunch of B and T cells that respond to all different antigens outside of the body. Okay? And as I said, each one of these responds to their own unique antigen, but because you have billions of these cells, you then have a big, large repertoire of antigens you can respond to. Now let's say you get infected with something, okay, and there's antigen present, and one of these cells recognizes that antigen. What that does is it activates that cell, and then that cell then goes through clonal expansion, where they literally make millions of copies of themselves. So now instead of having just that one cell that recognizes that antigen, you now have 1 million cells that can go off and fight the infection as it should. Okay? So without infection, you have just one cell that responds to each antigen. After infection, you get hundreds of thousands, if not millions of these that can then go off and fight the infection.
That's a question real quick. Okay, these cells that attack the cell, so each one, it might affect the liver, might affect the kidney, or whatever it is. So, okay, so they don't kill maybe the pancreas cells, that's why you get diabetes.
So it just depends on the antigen that they respond to, right?
Okay.
Why exactly it tends to happen more often to certain antigens than others, we don't fully know. But it probably has something to do with genetics. So the person just doesn't have the ability to express that antigen from these cells for whatever reason, right? But there are autoimmune diseases that just target everything. So lupus, for example, okay, or sarcoidosis. They go after tons of crap, right? And that's just because you got a whole bunch of these cells that escaped and they're just doing weird shit that they shouldn't do. So how do we prevent this? Again, autoimmune diseases occur when these cells escape, or the guys that respond to self-antigen. So they have to be destroyed, and this is a process called clonal deletion. Okay? Any cell that recognizes self-antigen is zapped and killed. Okay? If it does make its way out, then this autoimmune disease can develop, and this obviously can be problematic. So clonal deletion, this is self-recognizing clones. So these are the guys that recognize self. They are eliminated and destroyed, and that way they cannot be released and cause autoimmune disorders. Alright, so that is how the receptor is produced. Okay? It's a completely random chance event, right? Shuffling of some genes, slap them together, that's your antigen you respond to. Okay? It could be good antigen, it could be bad. If it's bad, they get destroyed during the maturation process, so that you're left with just a bunch of cells that respond to all other things. Okay? Now, what do these receptors actually look like? So when we're looking at the B cells have an immunoglobulin receptor. Immunoglobulins, also known as Igs, also known as antibodies. Okay? So really, B cells just have an antibody stuck in their membrane. And this is why they can bind to antigen as it's floating around. Because antibodies bind to things floating around. Okay? So here is the B cell receptor. It looks just like the antibody I drew on the board during the ELISA lecture. Okay? So you've got your 4 peptides. You have your FAB region up at the top, which has these variable bits, which can then stick to antigen. Okay? The FC region in this case is then stuck in the membrane of the cell, and this is what will tell the cell, hey, something bound to me, do something. Get ready. T cell receptors are similar, except they are not antibodies. Okay? And that's why they cannot recognize antigen floating around. Instead, it needs to be presented to them in MHC. Okay? So it looks like this. There's variable regions, and you can see these variable regions are actually inside of the receptor. Okay? And that's why MHC is required, because MHC will come in, open this up, and then this is exposed to the antigen. Okay, so those are our B and T cells and how they are developed. Again, this occurs in every individual from birth up until about puberty, and then it quickly drops off. Okay? Again, it has nothing to do with infection, and it's a completely kind of crapshoot random process. And this is why all of our immune systems are very different from one individual to the next. I may get the flu and it doesn't bother me. I give you the exact same flu when you're bedridden for 3 weeks. It has a lot to do with your genetics and just the random crapshoot of B and T cells you have, and whether or not you have enough of them to respond in the appropriate manner. Okay, so now we've got these cells. As I said, they just literally circulate around the body until you become infected. Okay? So now let's talk about what happens after the infection. So as we mentioned before, with T cells in particular, they cannot see antigen on their own. They need to see antigen in a molecule that is presented to them. Okay? Now antigens, as we mentioned before, is anything that can provoke or elicit an immune response. And these are most often going to be proteins or some sort of protein counterpart. Okay? They tend to be large molecules that are very complex in structure. Okay? This tends to give the best antigen response. If it's a large molecule that's not complex in structure, it's just like a single repeat over and over and over again, these tend not to work very well. Okay? So big protein with a bunch of random kind of bits associated with it gives you the best antigenic response. Now, as we said, these antigens need to be presented to T cells. Okay? This requires a concept known as cooperation in immunity. This is something that is a kind of an overarching trend that occurs again and again and again with all different immune cells. These cells have to communicate with each other. There's not just one cell in the immune system that does everything. Okay? T cells are very important, right? They help the immune response, but if it isn't for the antigen-presenting cell, they never become activated and they won't function. Okay? B cells work very good at making antibody, but without a T cell, they can never do it. So all these cells have to work together for it to function properly. You may ask why this is. Well, it's like this so that the immune system only gets activated when it's absolutely required. Okay? And that's good because if your immune system was activated all the time, willy-nilly, you would feel pretty awful. Okay? You'd have a lot of inflammation, you'd feel like you have the flu, and it could even be potentially life-threatening. So what is an antigen-presenting cell? Well, just like its name implies, it is a cell that presents antigen. Okay? These are phagocytes, so they phagocytose things, like bacteria, for example. They break it down during that phagocytosis, and then they put it onto the MHC class II molecules. Okay? The cells that are most often thought of as antigen-presenting cells are macrophages, dendritic cells, and B cells. Okay? And this is a little weird, but we'll talk about why that is later on. And again, they phagocytose or engulf antigen, break it apart, and then put it on the MHC class II molecule. Now this MHC class II molecule kind of acts like a hot dog bun. Okay? It's holding the antigen, which is the hot dog, and this can then be shown to the T cell, so the T cell can either recognize it or ignore it. Okay? Once the T cell does recognize that antigen, if the macrophage is lucky, then they communicate with each other by releasing chemicals or cytokines. Okay? The most prominent one is going to be IL-2. And when these chemicals are released between the two of them, it activates both of them. It makes them very happy, and then they go off and continue on fighting the infection. Okay? So how does this look in the real world? So we've got a macrophage in this case. It is binding to a bacteria, a microbe. This is then being internalized and being phagocytosed. Remember, during phagocytosis, there's lysosomes that fuse with this cell and break it down into small little bits and pieces. Okay? These little bits and pieces are now the antigens, and these antigens can then be put into the MHC class II molecule and put on the surface of the macrophage. Okay? The macrophage then will leave the tissue and actually go into the lymphatic system. And they sit there in the lymphatic system because that's where most of the T cells are. And they'll just sit there with it on the surface, and T cells will just come up and keep going around and around until the T cell that recognizes that antigen is found. And this is why sometimes it takes weeks for your immune system to fully develop. Because it's kind of a crapshoot. You've got 1 million cells circulating around, you got one cell sitting there, and they all have to go to it, right? You just got to hope that you find the one that responds. Once they do meet each other, so you have a T cell that recognizes the antigen, again, they're going to dump chemicals on each other, primarily cytokines, which then will activate these cells and make them function and fight the infection better. Okay. And I think it was you that asked before why your lymph nodes get swollen during an infection. That was you? Okay, someone up in front. This is a prime example of it, right? You got a macrophage sitting there, and you got all these cells coming around it and kind of swelling up the area. Okay. All right, so that is the antigen presentation side of things. Now let's talk about B cells and T cells and what they actually do. Okay. So B cells is what we'll start off with, and as we mentioned before, B cells are important in the production of antibodies. Now, the way that these guys work is they go through the same developmental process as T cells. So you have the random assortment to make your receptor that binds to a unique antigen. You then delete the guys that are bad, that are going to respond to self-antigens. And then they are released into the periphery. Okay? Now these cells are a little different than T cells because they can actually see antigen floating around. They do not need an antigen presenting cell. In fact, they can actually act as an antigen presenting cell if the case arises. Okay? So what tends to happen is, let's say I jab myself in the arm, I get inflammation, and I get all these cells recruiting to the area. After a little while, B cells will actually work their way into that area where the site of infection is, and then they will try to bind to the antigens that are present there. If they happen to bind to the antigen, they then internalize it through phagocytosis, process and present it on MHC class II, and they will present it to T cells. And the reason they do this is because B cells cannot do anything on their own. Okay? They can never become fully activated by themselves. Instead, they need a T cell to tell them that it's okay. So they have to process, they have to present to these T cells so that the T cell can go, okay, you're good to go and make antibodies. Once this occurs, the B cell is activated. They then make millions of copies of themselves, so what we call clonal expansion. And then most of these will become plasma cells, which will make a ton of antibody. And some will become memory cells, which give you immunological memory. So how does this look like in the real world? So up at the top, we have a B cell. You can see the B cell receptors all around it. These are antibodies. Okay? As I said, during an infection, the B cells will actually migrate their way into the infectious area. And then they will bind their antigen, or their antibody receptor to the antigen. They then internalize whatever is bound to the antibody, and then they phagocytose, process, and present it on MHC class II. They'll go back into the lymphatic system, they'll show it to all the T cells in hopes that they find the T cell that responds to it. If they do, they then dump a bunch of chemicals on each other, making them both activated. And an activated B cell will go make millions of copies of itself, most of which will make antibodies, known as plasma cells, and a small percentage of them will become memory B cells, which give you immunological memory. So what exactly is an antibody and why are they important? So as we mentioned before, antibodies are Y-shaped proteins, which are used to stick to antigen. Okay? They're often called immunoglobulins, or Igs. And as we talked about in lab, there's two parts to them. The upper part, known as the FAB, and the lower part, known as the FC. The FAB has these variable regions, which can stick to the antigen. The FC region, or known as the constant region, is the part that stays the same for all individuals within a particular species. So, the FAB region is where something called the epitope binds. An epitope is just a fancy word for something that binds to antibody. Okay? In reality, it's the same thing as an antigen, okay, at least for our purposes. There's small differences, but for us, epitope and antigen are pretty much the same. The FC region is what contains the effector portion. So this is what will tell whatever binds the antibody what it should do. Okay? How should it respond to it? How do antibodies actually bind to antigen? Well, it's very similar to like we talked about in Chapter 8 with enzymes and lock and key fit. It's all based on shape and charge. Okay? So here we have an antigen up at the top. It's got this kind of square shape. We have an antibody that is designed to bind to it, and it has a mirrored or corresponding shape to that square. Okay? It also has an opposite charge so that it sticks very, very nicely, kind of like a magnet. Conversely, if you have an antigen that is weird or different shaped, it's not going to bind and it will just fall right off. However, if you have an antigen that is similarly shaped, it may bind but very weakly. Okay, so it tends to fall off and not cause any problems. So what do antibodies do during an infection? Okay? Antibodies, generally speaking, are very important. They help clear an infection by doing 6 primary things. Okay? First and foremost, they tag cells that are foreign. Okay? In this case, they're tagging a bacteria. And all this tagging does is piss off the pathogen. Okay? It gets stuck on them, that slows them down, they can't move as much, it's harder to divide. All these things kind of slow them down and make it harder for them to infect you. Not only does this tagging slow them down, but it also makes them tastier to phagocytes. This is a process known as opsonization. Now when a phagocyte sees these antibodies on the cell, they freak out and they know right away, I'm going to eat that and I'm going to get rid of it. No questions about it. They can agglutinate. This is where they stick a pathogen together. This is why antibodies have two sticky parts, okay, one on each end of the Y. One can bind to one pathogen, the other can bind to another pathogen, and now they're stuck together. This tends to piss off the organism, making it harder for them to grow and divide. But it also makes it easier for phagocytes to come in and just gobble them all up at once. One big blob. They can fix complement, so we talked about this in chapter 14. Classical complement requires an antibody. As soon as the antibody is stuck to the cell, the complement proteins can form around it, poke a hole, cell dies. They can neutralize. This works really well for viruses. So viruses have proteins on their surface that they use to get into a cell. If an antibody covers up those proteins, the virus can't get in, and now your cells won't get infected. And lastly, they can precipitate. Okay? And this is where they bring really small molecules like toxins and venoms out of the bloodstream. So these very small molecules are floating around, they're very potent and can kill you very quickly. If you have antibodies to them, they will bind to these small molecules, making them heavy, and then they sink out of the blood. They can then be gobbled up by phagocytes, and now that toxin or venom no longer kills you. So these 6 things all together make antibodies very, very important. Okay? All this stuff together means that you either clear the infection or prevent the infection altogether. Now there are 5 different types of antibodies. Okay? IgG, IgA, IgM, IgD, and IgE. IgG over here on this side. This is the Y-shaped antibody that I drew on the board. This is by far and large the most common antibody found in the blood. Makes up about 80% of all antibodies in the serum. Okay? This is the kind of the best of all antibodies. It's the most ubiquitous. It can do all 6 of those things we just talked about relatively well. Okay? It is also the only antibody that crosses the placenta and can be transferred to your unborn child. Okay? This is the second antibody to be produced during an infection, and then it becomes the most abundant to be produced after. IgA, this is a dimer, and that just means it's 2 IgGs stuck together, end to end. This antibody is excreted primarily at mucous membranes, so areas of the body that are exposed to the environment. Okay? So your eyes, your mouth, your throat, your genitals, places like that. Its primary purpose is to bind antigen and pathogen, preventing it from infecting you, okay, altogether. This is also an antibody that can be transferred from mother to baby during the first couple days of breastfeeding, okay, in the colostrum. IgN is a pentamer, which means it's got 5 antibodies stuck together. Okay? This is the first antibody to be produced during an infection. All infections. And it is the best at agglutination and complement fixation. Okay? And you can kind of see why, because instead of just having 2 variable regions like in the IgG, there are now 10. So they can stick to 10 bacteria instead of 2. IgD is a monomer. It looks a lot like IgG, except it is stuck in a cell. IgD is the B cell receptor. You don't typically see this guy in the blood too often. And IgE is also a receptor, and this is the receptor for mast cells. We talked a little bit about mast cells in chapter 14. Those are cells that can cause allergic responses, but they also induce inflammation. Okay? And this is the receptor that they use to do so. And this is everything we just talked about in words, which should be useful for you in your study. So, antibodies are great at fighting an infection, but they're also great at preventing infection. Okay? And this is the basic concept behind immunological memory. It's the antibodies primarily that protect you from getting that infection for a second time around. Okay? So what we have here is a chart that shows the primary and secondary antibody responses. Primary antibody responses are what develop when you first get infected with something, very first time. Secondary antibody responses are what develop when you get infected with the exact same pathogen again, okay, or for a second time. So when you first get infected with pathogen, right, very first point, note that there's a long period where you are developing absolutely no antibodies, okay, to that pathogen. This can take days, sometimes weeks in certain cases. What's happening here is your body and your immune system are developing their response, okay? It takes a little while for, the neutrophils to come in, the phagocytes to gobble it up, to present it, to get the T cells and B cells activated, all that stuff, okay? It takes some time. So during that process, you're not making any antibodies yet. But eventually you will, and once these B cells go into overdrive, they start making IgM, as I mentioned before, is the first antibody to be produced, and then they switch over to IgG, and by the end of it, you get a pretty large blip of antibody that is produced. Now, these B cells that are making this antibody are known as plasma cells, and these cells are short-lived, okay? So once the infection is cleared, they die off, and you're left with just a very low level of those antibodies in circulation. Okay? But another cell was produced during this production of antibody. So not just plasma cells were made, but also memory B cells. These memory B cells, okay, don't do anything during this initial infection. Instead, they just kind of hang out, kind of bored, shriveled up, really do much, and just circulate around in the body. And they do that because they aren't going to respond to this guy. Instead, they're there to respond to this guy, the secondary infection. So once you clear the pathogen, the plasma cells die off, you have very low levels of antibody. But if you get infected with the exact same pathogen again for the second time, these memory cells will almost immediately recognize this, respond, and they make antibody very, very, very quickly in very, very high levels. Okay? So note now there's no longer this latency period or this little to no antibody production. Instead, almost immediately after exposure, you're getting a ton of antibody. And not only the same amount of antibody, but in fact more. Sometimes double the amount of antibody than what you received for the first infection. And this rapid response of these memory cells and their ability to produce this antibody very, very quickly is what prevents you from getting infected for a second time. This is what gives you that immunological memory response. These memory B and T cells are very, very long-lived. Okay? They can live for decades at a time. Okay? They circulate around the body, and they're just sitting there waiting for you to get infected for a second. So this is everything again. We just talked about in words. All right, so those are the B cells, the antibodies they produce, and how those antibodies are used at giving you immunological memory. Now let's talk about T cells. So T cells are responsible for something called cell-mediated immunity. Cell-mediated immunity is exactly as it sounds. These are cells that go around helping the rest of the immune system or other cells. Okay? A lot of immune cells in the body require involvement with T cells to function. So they're often kind of thought of as the brains of the immune response. If you don't get T cells to function, you really don't get an immune response because a lot of immune cells require them to be there. As we mentioned, T cells require MHC to see their antigen. They cannot see it floating around. Once they do see their antigen, though, they can then go off and stimulate all different types of immune cells, whether that be T cells, more T cells, B cells, phagocytes, whatever. And again, they primarily do this by releasing chemical signals known as cytokines. So how exactly do T cells become activated? Okay. So as we said, during development, you have a series of T cells that each respond to a unique antigen. Okay. And these cells will circulate around in the body, primarily in the lymphatic system. And they live for long periods of time, decades at a time, hoping to see their antigen. But remember, they cannot see their antigen floating around. Instead, it needs to be presented to them in an MHC molecule. So once you have an infection, antigen-presenting cells will gobble up that infectious agent. They will process and present it on their surface. They then go to the lymphatic system and they show it to all the T cells until you get a T cell that responds to that particular antigen. Now, if it is an antigen expressed in MHC class 2, it will be a CD4 cell. CD4 cells, also known as helper T cells, are the cells that go around helping the immune response, okay, as their name implies. They're the ones that go around activating B cells, other T cells, phagocytes, and so forth. If it's an antigen expressed in MHC class 1, then it's going to activate CD8 T cells. And CD8 T cells, also known as cytotoxic T cells, are the T cells that will go around killing any abnormal or virally infected cells. Okay? So it just depends on how the antigen is presented to them. Okay? If it's in MHC class 2, you get a CD4. If it's in MHC class 1, it's in CD8s. And as we talked about, MHC class 2 is used by antigen-presenting cells, which means it's going to be extracellular pathogens, like bacteria and parasites and so forth. Or MHC class 2 is found in class 1 is found in all nucleated cells, which are the cells that could potentially become infected with viruses or become abnormal for whatever reason. And so that's why these cells are the guys that will come in and kill them eventually. Once they recognize their antigen, they become activated. They then make many, many copies of themselves, and they go off and they either help if they're CD4 cells or they kill if they're CD8 cells. So T helper cells, or CD4 T cells, again, these are the cells responsible for helping the immune system. Okay? They're often considered the brains of the immune system because a lot of immune cells require them to function properly. So once a T cell or CD4 helper cell becomes activated, they make copies of themselves and then they go off and they go stimulate all different parts of the immune system so that it functions appropriately. Cytotoxic T cells, or CD8 T cells, do exactly as their name implies. Cyto meaning cell, toxic meaning death. So they go and they kill cells that are weird. Okay? By weird, I mean virally infected, having intracellular parasites like viruses or bacteria, or anything that's abnormal. These CD8 T cells will literally, once they become activated, make millions of copies of themselves, and then go to every single nucleated cell in your body, looking for that particular antigen. Once they find that antigen, they kill the cell. They kill the cell by releasing perforins and granzymes. Perforins make a hole in the cell, and granzymes enter the cell and cause something called apoptosis. Apoptosis is basically where a cell just shrivels up and dies. It's important that they cause apoptosis and not just cause the cell to explode open, because if a cell explodes open, it will elicit even more of immune responses, and that can cause shock. Okay? So this apoptosis, or shriveling up of cells, is very important so that the cells do not, or the dead cells do not cause more problems for the individual. Again, these work well against virally infected cells, cancer cells, and abnormal cells, but even cells from other animals or humans. Okay? So if I were to take your liver and put it into me, these cells would probably respond to it and destroy it. Okay? Because it's not from me. It was not my antigens that it was taught not to respond to. Okay? Now there are some exceptions to that. If you happen to have very similar types of antigens to me, then it would work. And that's exactly what they do when you go on an organ transplant list. They look for someone that has a similar match to you so that your immune system does not destroy it.
What does that analysis entail?
So it's looking at your HLA types. Okay, so HLA is MHC, right? There's two classes, but the way that they are configured genetically is broken up into 6 groups. Okay, and so you get 3 groups from mom and 3 groups from dad, and it's literally just a random 3 from either one of those guys. Okay, and so what they're looking for is at least 4 out of the 6 matches. Now, if you have 4 out of the 6 matches, you won't have a complete rejection, just a minor rejection, which they can control with immunosuppressive drugs. If you have a complete perfect match, so 6 out of 6, You put that organ in you and nothing happens. Your immune system just ignores it like it's nothing. But it's relatively difficult to do. It's kind of a game.
I have a question. If it does match 6 and 6, then that person will not need to be on immunosuppressant?
Correct. The most likely person, at least relative-wise, that will have a 6 out of 6 match with you would be a sibling, right? But it's still a crapshoot because you get 3 randomly from mom and 3 randomly from dad, and so did your sibling, right? So if you have 10 siblings, you might have a good chance. But if you only have 1, chances are it's not going to still be a big match. Okay, so how does this look in cartoon form? So at the top we have CD4 cells, at the bottom we have CD8 cells, okay? So both of these require antigen to be presented to them on an antigen-presenting cell, okay? So with CD4 cells, this is easy. These guys come in, they gobble up the pathogen, they process it, present it on MHC class II to T cells in the lymphatic system. They recognize the antigen, they make copies of themselves, and they go off and fight the infection, okay? Some of these copies will fight the primary infection at the time. Other copies will become memory B cells and kind of lie dormant and wait for you to get infected for a second time, okay? Just like in the B cells. CD8 cells, on the other hand, are a little bit different, okay? So this is why earlier I mentioned that antigen-presenting cells express both MHC class I and MHC class II. Remember, CD8 cells can only see MHC class I. And they can only become activated by an antigen-presenting cell. So the antigen-presenting cell has to express this protein, okay? And what a lot of antigen-presenting cells will do is they will actually infect themselves on purpose with the virus, so that the viral antigens get inside of them, they can process it, and then they can present it on MHC class I. In fact, HIV exploits this, and this is how HIV easily gets transferred from one cell to another. Macrophage gets infected by HIV, it's got HIV in it, T cell comes and talks to it, and every cell that comes and talks to it gets infected. Okay? Now, once this antigen-presenting cell gets infected by the virus, it breaks up its antigens, expresses it on the surface in MHC class I, then shows this to all the CD8 cells. Once it finds a CD8 cell that recognizes it, this activated CD8 cell makes millions of copies of itself, literally circulates around the body, looking at all nucleated cells, looking for that antigen. They find it, they then kill that cell, it shrivels up and dies. Okay. This is an example of a cytotoxic T cell, or CD8 T cell. Up here, you have some big-ass weird cell, and then you've got the little green CD8 T cell. Okay? This guy is tiny compared to this guy, but he kills it no problem. Okay? They can kill pretty much any cell in the body. Now, there are other cells that can kill, not just CD8 T cells. The other main type of killers are known as natural killer cells, or NK cells. These cells don't look for particular antigens. Instead, they look for really generalized differences. Okay? The reason this is important is because one way that viruses can survive in the body is they'll actually get rid of MHC class I. So as soon as they infect a cell, they get rid of MHC, and then a T cell comes by and it looks and it's like, oh, I don't see MHC, I'm going to leave. You're fine. Right? Well, natural killer cells can come by and they can look at it and they go, you know what, you're supposed to have MHC, you don't, you're weird, I'm killing you. So it's looking for generalized abnormalities, okay? Things that just you should not see in a normal, healthy cell. Okay, so that is the immune system. Now, let's go back and let's talk about it from start to finish in a brief kind of synopsis. Okay, I know there's a lot of detail. We'll go back to our initial little schematic that I skipped. And this is broken up into two parts, the orange part and the blue part. Okay? The orange part is the part that occurs in all individuals, okay, from birth up until puberty, and this is the development of those B and T cells. Okay? Remember, B and T cells are produced via hematopoiesis, okay, in the bone marrow, and then they need to be matured in the thymus or bone marrow, thymus if you're a T cell, or bone marrow if you're a B cell. And during this maturation process, you are getting rid of any cell that responds to self-antigens. Okay. These guys are then released into the periphery, and then they can circulate around the body waiting for an infection. So now that brings us to the blue part. This is the infection. Okay? Now this blue part on the board is only describing what's happening with the the adaptive immune response. But let's take it a step back and talk about the innate immune response as well, because that is what gets the ball rolling, okay, for all of this to occur. So let's use my example I used last class. Okay, I've got a needle, it's full of E. coli, I inject it into my arm. Does anyone remember the first thing that's going to happen? Inflammation, right? So I inject the needle into my arm, that kills, breaks tissue down, and that causes inflammation, right? What does inflammation mean? What's the primary sign of inflammation? Redness, swelling, edema, right? So buildup of fluid. And what does this inflammation do? Exactly, it recruits cells to the area, okay? And who are the first cells to come to the area? The garbage trucks, neutrophils, right? They come in and they gobble up stuff and then quickly die. However, if they don't get it under control, then other cells come to the rescue, okay? Those are primarily antigen-presenting cells, like macrophages, or B cells, okay? Now that brings us to our part, the blue part, okay? So we've got pathogen there, it's not being cleared by the neutrophils, so now we've got macrophages coming in and B cells coming in. Macrophage comes in, it sees the antigen, it gobbles it up, breaks it apart, and presents it on what molecule? MHC class 1 or 2? 2. Okay? So with antigen-presenting cells, unless it's a virus, it's going to be MHC class 2. Okay? So, gets expressed on MHC class 2, the macrophage then goes back into the lymphatic system, and it shows it to all the T cells it sees. Okay? Now, it's hoping to find that one T cell that recognizes that particular antigen. Once that happens, they then dump chemicals on each other, they all become happy, right? The T cell then makes many, many, many copies of itself, okay? And these can then either go off and fight the infection by either helping or destroying, okay, cells that are bad. Or they can become memory T cells that circulate around the body waiting for a second infection. Okay? So there's the T cell side of things. Simultaneously, the B cells are also going through a similar process. Okay? This is why your book has a line down the middle and they're going kind of at the same time, because it's literally what is happening. So macrophages are gobbling up material, but also B cells are there looking for antigen. And if the antigen then sticks to the B cell receptor, they also phagocytose, gobble it up, and present it on MHC class 2. They then go into the periphery, into the lymphatic system, and they show it to T cells in hopes that they find that T cell that recognizes the antigen. If they do, the T cell then activates the B cell. If the T cell does not activate the B cell shrivels up and dies. Okay? It needs the T cell to function. Once it becomes activated by the T cell, then makes millions of copies of itself, most of which will become plasma cells that pump out antibody, and a small fraction of which will become memory cells that then sit there, lie in wait for a second infection. Okay? So that is chapter 14 and 15 in a nutshell. Okay? There will be an essay question on the immune system, just giving you a heads up. And it will be very general, and it will be what happens during an infection type of question. Okay? So make sure you have at least a running idea of what's going on through these processes. Okay? Alright, so that is the immune system. How's everyone feel about it? So what I just taught you was like a semester's worth of material. Okay, so I don't expect you to know every single detail. Now, I didn't go into a lot of details. I skimmed over a lot of material too, okay? But I do want you to know the gist, okay, what we just talked about. I also want you to know any bold words in here, okay? And that's usually a good kind of take-home message, because those are the words I like to pick for the matching, if you haven't noticed already. Okay. Lastly, what we're going to talk about is